Bismillahirrahmanirrahim. Now our topic of discussion is ketone body synthesis. In last lecture, we have studied regarding the metabolism of fatty acid. The ketone body synthesis occur when there is glucose level is low. So in this diagram you can see one important thing that this is acetyl coenzyme. This is intermediate of our all metabolism, like protein, polysaccharide, lipids. If you understand, if you make a sense of this mind map, you can describe any metabolism, any cycle regarding the metabolism. So when there is glucose level low, so acetyl coenzyme A, this acetyl coenzyme A can't go into citric acid cycle. So this cycle stopped because oxaloacetate, which is intermediate of citric acid cycle, start the process of gluconeogenesis to maintain glucose level. So then acetyl CoA start another cycle, which is ketone body synthesis process. Now let's discuss ketone body. When use of fatty acid in citric acid cycle require carbohydrate for the production of oxaloacetate. Because we are in condition of starvation or in condition of diabetic person which is unable to intake carbohydrates, because we require carbohydrate for the oxaloacetate formation to start our citric acid. But in case of starvation or diabetic person with patient is unable to produce oxaloacetate to make glucose level is low, so oxaloacetate start gluconeogenesis. So in this way the citric acid cycle stop down. Now the other mechanism start ketone body synthesis to meet our energy level. In this diagram you can see acetyl coenzyme A is converting with the help of oxaloacetate is converted into citrate, but when there is low glucose in condition of starvation or diabetic person, gluconeogenesis started, so citric acid cycle stopped down. Now the other pathway for the fate of acetyl CoA is ketone body formation synthesis. In this corner you can, and very important concept is that fatty acid cannot be used to synthesize glucose. Glucose has ability to store in form of fatty acid, but fatty acid have not able to synthesize glucose, mean they cannot start glucose. Although fatty acid can generate acetyl CoA, but it is unable to generate glucose. So keep in mind this is very high yield concept. Now let's discuss how ketone body is formation. On this side you can see these are acetyl CoA, one acetyl CoA and second acetyl CoA. They start to make acetoacetyl CoA. Two acetyl CoA combine to form acetoacetyl CoA. These are two carbon. These are also two carbon. Now it is 4 carbon molecule. You can see that 1, 2, 3, 4. 4 carbon are present. In this is 1 carbon, 2 carbon, 3 carbon, 4 carbon. Now with the another acetyl CoA that is here, now it converted into 6 carbon, 3 hydroxy, 1, 2, 3, 4, 5, 6 carbon. Now it converted into 6 carbon, that is hydroxy, 3 hydroxy, 3 methyl glutaryl CoA, or you can see HMG CoA in simple form, HMG CoA. So acetyl CoA to acetyl CoA combine to form acetoacetyl CoA, which is 4 carbon. With the third acetyl CoA, they form HMG CoA. Now it's step to form our ketone body. First, let me change the first, that is acetoacetate formation by the release of this acetyl CoA. In the third step, now 4 carbon acetoacetate is formed, which is acetone, but it is a beta ketone, because the keto group is present on beta on this carbon. So in this way, it is the important concept regarding the beta ketone. They are very unstable because they break down spontaneously into acetone by decarboxylation. The CO2 is removed in this phase, so now 3 carbon acetone is formed. And other conversion, if NADH is present, it provides them hydrogen. Now it converted to 3 hydroxybutyrate. This hydrogen attached to this negative oxygen. So now it is dehydroxybutyrate. So all these 3 are acetone, which are formed with the help of acetyl CoA. So 3 ketone bodies: acetoacetate, acetone, D3 hydroxybutyrate. So these 3 ketone bodies are formed. But another concept, the acetone has fruity odor. When the ketone body utilizing person breathe, we can smell the acetone. They have fruity odor in their breath, and they also maybe have ketone body in their urine. So the acetoacetate and beta hydroxybutyrate. So acetone simply goes out, but the acetoacetate have ability to convert into 2-acetyl CoA, which provide us 23 ATP, and beta hydroxybutyrate give us 3 ATP. So this is the mechanism regarding the production of ATP from our ketone bodies. Now another important concept, that liver had made ketone bodies, but it is used in extrahepatic tissue, mean they are not utilized by liver, and other extrahepatic mean it may be a brain, it may be a muscle, or any other tissue, peripheral tissue. So now acetoacetate come into our muscle, or 3 hydroxybutyrate come into our muscle. They provide NAD to convert the NADH. In this way NADH is obtained, and acetoacetate with the help of thiophorase enzyme, which is present in only peripheral tissue, it is absent in liver. So in this way we've got acetoacetyl CoA with through the TCA cycle, and finally succinyl CoA is obtained in TCA cycle, are 2-acetyl-CoA, which can be utilized by TCA cycle for the production of ATP. But another important concept is that we already told that oxaloacetate is deficient, but it is deficient only in liver. In peripheral tissue, gluconeogenesis is not. Glucose is only limited to our mainly liver. So in peripheral tissue, a lot of oxaloacetate is available. So this acetyl-CoA has ability to jump into TCA cycle to provide ATP. So remember, very important, that thiophorase enzyme is absent in liver. Thiophorase is absent in liver. That's why ketone body cannot be utilized by liver. The liver is major source of ketone body. They have ability to produce ketone bodies, but it is transported into blood to other tissue, extrahepatic tissue. It is transported to extrahepatic. It may be a brain, it may be a muscle, or any other extrahepatic. Extrahepatic means excluding liver. Acetoacetate in tissue, first aceto is activated to acetoacetate by transforming CoAs from succinyl-CoA enzyme. Then it's split into 2-acetyl-CoA by the enzyme of thiolase reaction. In this step, the thiolase enzyme is used to split the acetoacetyl-CoA into 2-acetyl-CoA. So now a pathological condition, last discussion regarding the ketone body. Ketosis means high level of ketone. When a diabetic person is not utilizing enough ketone body, mean they are producing more but utilizing less. The production of ketone body is higher than the utilization. In this case, ketone bodies become higher in blood. That condition is ketonemia, which lead to acidosis. And other problem, because these blood is also circulating through our kidney, so in that way ketone body also increase our urine. So in this way, that condition is called ketonuria. So that's lead to dehydration. Why dehydration? Because they are osmotic, mean they are decreasing our osmotic level. So in that way, they also lead to dehydration. When the person in condition of starvation, so it's necessary to utilize water, drink water to compensate this dehydration. And acidosis, may they have pH level low pH because they are losing ketone. In this way, they lead this problem. Now it's complete our discussion regarding ketone metabolism, which is very high conceptual, but I have make it easy.